I'll be presenting on the topic Hydrocarbon-degrading Fungi at the Siljan Impact Crater, which was also my master thesis, supervised by Dr. Magnus Iversen, conducted at the Swedish Museum of Natural History and Micromine with the support of Erasmus Mundus program. So, when we think of impact craters, we often think them to be so destructive. Take, for example, the Chicxulub crater in the Yucatan Peninsula, Mexico, which happened around 66 million years ago, wiping off the dinosaurs from the face of the earth. But this need not to be the case always, as they can also provide favorable environments for microbial life. So once an impact event happens, the ground beneath gets heated up so much that it results in magma formation as well as these cracks and fractures, and these then serve as spaces through which water and minerals can seep through. Together, they form something called as a hydrothermal system. We might have heard of it, like these includes these hot springs and geysers, etc. The hydrothermal system then can be favorable environment for microbial life that is deep down in the deep subsurface, which is a place that most of us misunderstand to be a place where there is no life at all. But recent and many past studies have shown that life exists deep down, and these fractures can then be a new habitat for them. So these include thermophiles, which are the heat-loving microbes. Then we have the chemolithotrophs, which uses chemical reactions for their source of energy. Example being the sulfur oxidizing bacteria. Then we have anaerobes, which does not require oxygen for their survival. Take, for example, the methanogens, which can use carbon dioxide and hydrogen to produce methane, and that is why they're called methanogens. So once the impact event begins to cool down, these fractures gets filled up with minerals, and the organisms or the microbes that are present within gets preserved as fossils. There are a lot of studies on such impact related fossil life. Take, for example, this one from Siljan Impact. So these fossil microbes got trapped within these fractures, but they were using the energy from the hydrothermal system caused by the impact event. But our focus was the Siljan Impact Crater, again in Sweden. But this was formed around 380 million years ago. So when we're talking it in a geologic timescale, it was formed during the Devonian period. So this is much, much older than the impact event that wiped off the dinosaurs, which is pretty recent, just 66 million years ago. But what's most interesting about Siljan are the reports of seeping methane and oil, which has been known from 1730s and still exists today. So we might be wondering, how can an impact event lead to oil and gas formation? So millions of years ago, the area where Siljan is located now was covered with ocean. So in the ocean, we know that there's life, right? Oceanic life, be it plankton, algae. So once these oceanic life dies, they get settled to the bottom of the bed or bottom of the ocean. So there will be more organic matter deposition, more organic matter deposition. Since there is no oxygen deep down, it does not decompose. It just accumulates and convert into a mud-like structure. And eventually, all these organic mud gets condensed and clumped together to form a rock. And the water, since because there's like a lot of pressure, the water oozes out, and this condensed rock forms something called as shale. The more organic matter that shale has, the more black it appears. So we have black shale, which represents more organic matter. So when the impact event happened in Siljan during the Devonian period, 380 million years ago, the asteroid hit these shale-rich region, and the heat during the impact heated up the shale so much that it started producing oil. So the organic matter is a complex mixture of hydrocarbons. So hydrocarbons means CH bonds. So these complex CH bonds get broken down into simpler CH bonds like ethane, methane, propane, and that's why we have oil and gas production in Siljan. There was an interesting study uh, on fossil fungi that was found within the Siljan Impact Crater. But what's most interesting is that the scientists also found that these fungi were supporting methanogens to produce methane. So we might be thinking, where does this fungi get their source of energy from? How does the methanogens use the substrates that are produced by the fungi? So the short answer is the oil, the hydrocarbons that we just talked about. So my aim of the thesis project was to see that if there are any living fungi within the Siljan oil. We know that there are fossil fungi, but are there any living fungi within the oil? And then if there are, what is their potential to degrade these hydrocarbons or the CH bonds, thereby producing substrates like carbon dioxide or hydrogen to the methanogens to produce methane, and then to discuss the astrobiological implications of using Siljan as an analog to explore impact craters as interplanetary habitats and long-lived biological systems through this fungi-driven carbon cycling method. So my fieldwork was conducted at the Solberya quarry, which is again located within the Siljan Impact Crater. We could actually see oil seeping from the walls of the quarry, which is pretty cool. And then we collected these oil samples from drill holes that were present throughout the quarry, and we poured them into these bottles and tubes. And I also took my small field notebook to note down some points as well. Over the course of time, it was around 6 months altogether. We had a lot of experiments that were conducted, like sending the fungal cultures for identification, observing their microscopic features, blah blah. But I don't want to bore you guys with all those information, but I just want to focus on this particular experiment, which is called as DC-PIP experiment. I'm not going into too much technical stuff, but it's nice to know that there is this technique wherein we can know if the fungi is actually degrading the hydrocarbons or the oil. So what happens here is that we have this redox indicator called the CPIP. It is blue in color when it comes in this bottle because it is oxidized. And then we add the sipip to the flask, we add the fungal isolates, we add some yeast extract so that we provide some nutrients for the fungal isolates to grow, and then we add some crude oil. An important point here is that we did not add we didn't add the same oil that we collected from the field, but instead we added refined crude oil, because the Siljan oil itself in its natural form is like so complex. It has a lot of complex hydrocarbons like long chain alkanes, alkenes, and then benzenes, paraffins. So it's all very complex. But the refined crude oil will not have such complex system of hydrocarbons, so that it will be easy for these fungal isolates to use them as a source of energy, so that we don't exhaust them. We want them to grow. And then the process begins. We just left them undisturbed. So if the fungal metabolism is active and they use the hydrocarbons or the CH bonds that is present in the oil, they start breaking them down. The longer CH bonds break them down into shorter CH bonds. So this process will release electrons. And these released electrons then will be accepted by the dissipip, which is oxidized and gets reduced. So when it gets reduced, it turns colorless. So that is an indicator for a clear-cut hydrocarbon degradation taking place here within, with the fungi. So these are the results of the different isolates that we obtained. We send them for molecular identification. And we also observed some microscopic features. We can classify them up to their genus level with the help of microscope. Like there's this sporangia, which is typical of Apsidia, and these brush-like appearance is a typical feature of Penicillium. But it's not possible to identify up to their species level just with the microscope. That's why we did the molecular identification. Now here comes the most awaited results of the dissipip. So as you can see, Penicillium citrosulfuratum, this particular species had the fastest metabolic activation of hydrocarbons, meaning they turned colorless within 1 week of the experiment. So this means that they were able to break the carbon hydrogen bonds present in the oil, use the electrons. That's why we have the transition from dark blue to colorless. So here we have the control sample which does not have any oil added to it. And we also had other controls as well, wherein we had just yeast extract, the disipep, and the isolate, and it did not change colors. So it means that the fungi being in heterotroph, it requires a source of energy in order to get, or in order for it to activate its metabolic processes. So let's put everything together. What is happening here? So we know that the impact even itself hit the shale and it heated up the shale, resulting in oil production. So the fungi can use this oil for their source of energy and break these complex hydrocarbons or the CH bonds that are present with alkenes, just as I just mentioned, into shorter hydrocarbon chains. And finally, the end products will be carbon dioxide, hydrogen, water, etc. The shorter ones, the shorter versions. Now, when conditions becomes anaerobic, methanogens uses these substrates for their biologic pathways. So we just mentioned that methanogens uses carbon dioxide and hydrogen for their methane production, which is just one pathway, which is called as a hydrogenotrophic pathway. They also uses other substrates as well, but we have just focused on one particular pathway here. So when conditions becomes anaerobic, since they're anaerobes, methanogens can use them for their biologic processes. This leads to my final illustration of comparing Siljan to the Gale crater on Mars. It doesn't seem that obvious at first, but let's look into it. So in case of Siljan, we know that the impact event heated the shale, which is now, resulting in oil production used by fungi, and then the substrates we have here. So the methanogens uses the substrates using this hydrotrophic pathway, which is the chemical reaction that has been shown here to produce methane. On Mars, we're not saying that there's fungi on Mars, but instead there could be putative Martian heterotrophs because we might be also thinking, why can't methanogens directly feed on the oil and gas for getting their substrates? They cannot do that. They need an intermediate character for them or to make the substrates bioavailable to them. So that's where fungi plays a role. So on Mars, there could be such similar heterotrophs which can be feeding on organics delivered through meteorites. So meteorites can also deliver hydrocarbons and organics. Now, if life was ever present on Mars, it could have been similarly keratinized and converted into shale and oil, possibly, and then which can also serve as the source of energy for these fungi. And then there could be these methane producing microbes, since we have a lot of reports of methane spikes on Mars and seasonal variations of methane, which suggest a possible microbial origin for it. There could be, we may never know, which can be producing methane. And this produced methane can then be stored in clathrates. And there was this interesting article that I was reading about, which mentioned that once the rovers move through the craters, it results in destabilization of these clathrates, which can result in producing methane, which can be one possible explanation why we have methane spikes on Mars. So this was just a study discussing the potential of impact craters to be long-lived biological habitats, because once the impact-induced hydrothermal system ceases to exist, these microorganisms shift to alternative sources of energy. For example, in case of Siljan, once the hydrothermal system uh, ceased to exist, they started depending on hydrocarbons from the oil for their survival. And there is this symbiotic relationship between different microorganisms, and together there's this whole biological habitat that is existing in Siljan. So this can be also a possible scenario on Mars or on other planets. We may never know, but it was just discussing the possibilities. So I hope you guys enjoyed my presentation. If you have any questions, please do let me know. Thank you so much, and have a nice day.